Parkinson's disease is characterized by the physical degradation of the brain's motor control centers. The incidence rate of this disease is almost twice as high in men as it is in women. Schizophrenia, a severe psychiatric disorder, typically manifests in a patient's early 20s. However, women exhibit an anomalous epidemiological pattern. They frequently experience a distinct secondary peak of onset delayed well into their late 40s and early 50s. This visualization shows 3 epidemiological trend lines, including a third discrepancy found in substance use disorders. Women exhibit a telescoping effect. Telescoping is a clinical phenomenon where a patient progresses from initial substance use to severe physical dependence at a significantly faster rate than average. Motor tremors, delayed psychotic breaks, and rapid chemical addiction present as completely different symptoms. Yet all 3 conditions are driven by a single neurochemical failure, dopamine dysfunction. For decades, neuroscience research operated on a specific assumption. It treated the human brain as a sex-neutral entity, establishing a default biological baseline that supposedly applied to everyone. The clinical data requires a different conclusion. The dopaminergic architecture is highly sexually dimorphic. The human species operates on 2 distinct structural models for dopamine synthesis, release, and learning. Applying a single neurochemical model to both sexes guarantees high rates of misdiagnosis and ensures ineffective treatments for half the population. This 3D model shows the standard textbook dopaminergic architecture. It highlights the mesolimbic pathway, which governs reward, and the nigrostriatal pathway, which controls voluntary movement. If we zoom in to a single synapse, we find the baseline cellular assembly line. The rate-limiting step here is tyrosine hydroxylase, or TH. TH is an enzyme that acts as the catalyst to convert dietary amino acids directly into dopamine molecules. Once synthesized, a protein called VMAT2 packages the dopamine for delivery. VMAT2 is a specific transporter that scoops the neurotransmitter up and seals it inside microscopic cellular vehicles called vesicles. After the vesicles release their contents across the synaptic gap, the signal must be cleared to maintain precision. The dopamine transporter, or DAT, vacuums the remaining dopamine back into the original neuron for recycling. DAT is a membrane-spanning protein dedicated to clearing the synapse. The mechanics of this assembly line are highly documented, but the assumption that this pipeline operates identically in both men and women is biologically false. During adolescent development, the male brain solidifies its baseline. Hormones like testosterone and its metabolite, DHT, act as structural engineers, locking in a rigid, high-capacity neural pipeline. Testosterone drives this organizational phase by directly upregulating the genetic expression for the VMAT2 packager and the DAT cleanup crew. This heavy structural investment primarily builds out the nigrostriatal pathway, preparing the motor circuit for high-volume signaling. This animated diagram illustrates the result, an amplitude-driven network. When a male brain encounters a rewarding stimulus, the system is designed to release a massive surge of dopamine, flooding the synaptic gap. To prevent these huge chemical spikes from overwhelming the circuit, the male brain compensates by heavily modulating inhibitory D2 receptors. D2 receptors are a class of dopamine sensors that act as the network's brakes, dialing down neuron activity to maintain strict homeostasis. This setup, large release volumes paired with heavy inhibitory brakes, dictates behavioral output. The massive stimulus-evoked dopamine release in the ventral striatum strictly correlates with high motivation, competition, and effort-heavy reward-seeking. The male architecture utilizes a high-output brute force neurochemical strategy. It trades flexibility for stability and raw signaling power. The female baseline operates under entirely different parameters. Here, estrogen acts as a dynamic conductor, orchestrating a highly plastic network designed to pulse and reorganize rhythmically across time. The primary female sex hormone, 17-estradiol, operates simultaneously across multiple timescales. In milliseconds, it binds to cell membranes to rapidly alter neuron firing rates. Over days and weeks, it directs gene transcription inside the nucleus, instructing the cell to alter its production of TH and DAT proteins. The physical wiring diverges here as well. Anatomical studies show females possess larger and more numerous dopamine neurons originating in the ventral tegmental area, the pathway dedicated to reward and cognition, rather than the motor-heavy nigrostriatal pathway dominant in males. This physical difference extends to the network's sensors. Healthy female brains exhibit a higher baseline density of D2-like receptors outside the striatum. This places far more listening posts in executive areas like the prefrontal cortex. Because estrogen directly drives dopamine synthesis and release, the entire network is cyclical. A woman's baseline dopamine activity rises and falls in direct synchronization with her hormonal dynamics, peaking precisely when estrogen levels are highest. The female architecture is efficiency-driven. It relies on extreme receptor sensitivity, dynamic modulation, and rapid neuroplasticity to achieve results, rather than raw chemical amplitude. Comparing these two models exposes a contradiction in reward learning data that violates standard biological rules. In condition-response trials, female subjects consistently exhibit more robust, highly motivated behavior to reach a reward cue than male subjects. This split-screen chart illustrates the contradiction. On the left, the male brain produces a massive dopamine spike to generate a moderate behavioral output. On the right, the female brain pairs a minimal dopamine release in the nucleus accumbens with maximum behavioral drive. According to the universally taught model of synaptic plasticity, neural connections require massive dopamine surges acting on NMDA receptors to learn. Under that standard framework, achieving higher behavioral output with a smaller dopamine release is biologically impossible. In the female brain, synaptic connections bypass the NMDA receptor. Instead, an L-type voltage-gated calcium channel and an estrogen receptor alpha link together. Estrogen receptor alpha, or ER-alpha, is a specific protein that allows cells to detect and alter their function in response to estrogen. This combination forms a completely distinct NMDA-independent learning pathway. When estrogen is present, this unique receptor complex fires a strong cellular response, allowing the brain to hardwire reward behaviors without ever requiring the massive dopamine surges seen in males. Male and female brains achieve the exact same behavioral learning goals by utilizing entirely distinct cellular machinery. With these two architectures defined, the anomalous clinical data from earlier perfectly aligns with synaptic reality. Parkinson's disease primarily destroys movement centers. The male brain is highly vulnerable here because its physical output relies entirely on the structural integrity of that high-capacity nigrostriatal pipeline. When those dense conduits degrade, the motor system fails. This graph tracks the female dopaminergic reserve. Because women start with a higher baseline density of DAT cleanup proteins and constant estrogen modulation, their brains buffer against initial neuronal loss. But when estrogen production plummets at the menopause threshold, that protective reserve collapses. That abrupt withdrawal of the estrogen buffer removes the network's neurochemical shield. This single biological event dictates both the delayed onset of Parkinson's in older women and that delayed secondary peak of schizophrenia incidence. In the context of addiction, the efficiency-driven female architecture becomes a severe vulnerability. Estrogen's capacity to rapidly escalate dopamine neuron firing rates directly drives the telescoping effect in substance use disorders. Estradiol preferentially boosts dopamine release in the dorsolateral striatum, the brain region dedicated to habit formation. This specific interaction rapidly converts the initial voluntary action of taking a drug into a hardwired compulsive behavior. Fluctuating hormones and dimorphic wiring serve as the strict biological determinants controlling disease onset and severity. Failing to map these differences causes direct patient harm. Women with Parkinson's consistently suffer higher rates of severe involuntary muscle movements from the primary treatment drug, levodopa, because standard dosing is calibrated to a high-capacity male baseline. The diagnostic criteria for childhood ADHD systemically miss young girls because their D2 receptor pruning trajectory is gradual. It does not produce the intense, highly visible hyperactivity generated by the male receptor pruning cycle. Treating the female brain as a modified variation of a default male standard guarantees clinical failure. The next phase of pharmacology requires targeted therapeutics. Here, a selective estrogen receptor modulator, or SERM, is locking cleanly into an ER-alpha receptor. A SERM is a class of drugs that can selectively block or activate estrogen receptors in specific tissues, offering precise psychiatric intervention. The discovery of non-canonical female structures, like the NMDA-independent learning pathway, provides entirely new maps for drug discovery. We can now target the exact cellular machinery unique to the female brain. medical research must integrate rigorous lifespan tracking. We have to measure precisely how these distinct dopamine circuits reorganize themselves during puberty, pregnancy, and menopause to preempt neurological decline. We have the biological data to permanently discard the sex-neutral model. True precision medicine begins when we map the dimorphic brain, leveraging these distinct architectures to engineer effective health outcomes for everyone.
Thanks for watching. If you learned something from this video, please subscribe to the Concierge Care channel. You can find us at health.conciergecare.net. Follow our links to other videos on deeper dives into the selected topic and related articles on the discussed subject. Don't forget to enable notifications so you don't miss our next uploads and live events.